morning, everyone in Dubai. Japan is currently at 15 noon now. Well, the main subject is I wanted to make a presentation in Dubai, but I'm concerned about the spread of infection these days, so I will do my online. So we would like to thank you for giving us the opportunity to make a presentation in spite of the different areas, because my major is cosmetics, so it's too different from your conference. So thank you very much. So this time we talk about clinical evaluation after introduction of stem cell supernatant Biocell Shot 65 by electroporation. So first, let me briefly introduce you to us. We are a company that conduct 3 items: research, and manufacturing, and clinical evaluation. And in this research, we are verifying the effect of introducing the stem cell culture solution called Biocell Shot 65, developed by our company into the skin. This is introduction. So there is a clinical report about umbilical cord blood-derived stem cell treatment for patients with psoriatic arthritis, the result of which was beneficial. Margaret et al. Concluded that stem cell is in umbilical cord blood support tissue repair and suppress over inflammation. There is another report about tissue repair support and over inflammation suppressing by umbilical cord blood derived stem cells. It is well-known fact that umbilical cord blood derived stem cells have some effects in the body. Cells in the body produce many cell growth factors for self-growth, cell migration, and immunosuppression. Various treatments by cell growth factors are applied in clinical and aesthetic practice based on the principle. And in our study, we used umbilical cord blood-derived stem cell culture supernatant, which is rich in TGF-beta. TGF-beta is involved in the wound healing process, such as inflammation, angiogenesis, fibroblast growth, collagen synthesis and deposition, and extracellular matrix remodeling. Umbilical cord blood-derived stem cell culture supernatant also contains platelet-derived growth factor, we call it PDGF, as a main ingredient, which activates many metabolic processes such as synthesis of protein and collagen, and collagenase activity, migration of my fibroblasts and smooth muscle cells. And since TGF-beta and PDGA are expected to improve skin, such as age spots, wrinkles, and pores, we examined the effect of cosmetic Biocell Shot 65, in addition to application of culture supernatants to the skin. In this study, introduction to the skin by electroporation was performed. This is the machine of electroporation. The results were evaluated with the facial diagnostic device during the 3 months after the start of the procedure. This is the diagnostic machine. We call it high-mirror professional. This is the Biocell Shot 65. Biocell Shot 65 contains more than 300 growth factors. In contrast, the usual agents contain only a single growth factor like EGF. EGF is widely used in China as a burn. So Biocell Shot 65 also contains EGF. A single application of EGF to the skin promotes the growth of epidermal cells and the fibroblasts and the production of vascular and cellular growth factor and hepatocyte growth factor. Biocell Shot 65 is expected to affect the skin in various ways because it contains many growth factors which have beneficial effects on the skin even with single factor. In particular, TGF-beta1 and PDGF-AA are contains a lot. So they are expected to have remarkable effect. TGF-beta1 promotes production and the deposition of extracellular matrix and has an anti-inflammatory effect. PDGF also acts as a modulator and is expected to suppress cell overgrowth and tumor development with beneficial effects to modulate other growth factors. Therefore, we introduce this solution to the patient, only 1 milliliter per application. In addition, in order to carry out the introduction more smoothly, we use Celemore Potele. It is a lotion contains collagen. Was used in combination as a solvent for Biocell Shot 65 at the same time. This is a machine of electroporation. The name is Bebe Pinocchio. Pores in cell membrane open, so various substances can infiltrate into the skin cells. Suzuki proved that hyaluronic acid molecular weight 1 million penetrates the skin with the device. Therefore, the ingredients stem cell supernatants can be introduced into skin cell with the device. In Japan, we use it in clinic or este. This is a face diagnostic device. In this study, stains, wrinkles, fine lines, pores, and foveae were evaluated. And maybe foveae is a specific facial area measured from front photo of the face. By measuring this area, the facial proportion is quantified. It is expressed by zone V1, where the center is the cheek. At zone V2, where the center is the side of the mouth. The ratio determines the facial proportion. The cosmetic results were conventionally evaluated by impression, but foveae is one of the markers which quantifies the cosmetic result. This is V1, and this one is V2. 4 aspects for assessing the degree of deterioration: improvement of face proportions. Of the 4 faces, the 2 on the upper cheeks are V1, and the 2 on the mouth to the lower cheek are V2. Together, it is called foveae. The patients spend their daily life in the usual way without diet or sleep restrictions. Patients were extracted from the patients visiting the clinic. Since the Biocell Shot 65 used its cosmetic product, The experiment was performed according to the guidelines established by Purecode, which means our company. The patients were selected from those who agreed to the experiment among the patients who visited the clinic and those who are healthy and had no serious illness. If abnormal reactions such as skin redness or observed during the trial, it was planned to discontinue it. Let's talk about significant difference. So this is a result. It showed the error from the ideal value set by the diagnostic machine, and the smaller the value, the closer to the ideal value. So in patient when 1, improvement was confirmed in wrinkles and fine lines around the eyes and stains and pores after the first treatment, but no difference was confirmed in 4V. Macroscopic findings confirmed improvement in dullness and wrinkles in the eyes. The patient 2 improvement was confirmed in fine line around the eyes after the first treatment, but no improvement in other items was confirmed. Also, no changes in macroscopic findings too. In patient 3, no improvement was confirmed in all items. If the value is close to ideal value before the treatment, it is considered difficult to confirm the improvement. So in patient 4, improvement was confirmed in wrinkles and also fine lines around the eyes and stains and pores after the 5th treatment. So in average, improvement was confirmed in wrinkles and the fine lines around the eyes and the stains and the pores. In conclusion, for we cannot any changing. We cannot find any changing. And wrinkles and fine lines around eyes, stains, pores, we find improvement. This is the reason. 4V, about 4V, no improvement was confirmed because 4V does not improve unless an approach is applied to the muscles. And about wrinkles to pores, although there was variance in the degree of improvement, the items were expected to improve due to simple application and deep introduction by pollution, so they are improved steadily. In addition to application of cultured supernatants to the skin, introduction to the skin by electroporation might improve stains, wrinkles, and pores. Our evaluation was performed with a facial diagnostic device during the 3 months after the start of the procedure. Thank you for listening. And if you have any questions, please contact us at the following email address. Thank you.